Just wait for you to tell me when to go.
Be a quick welcome at least.
Okay.
So, hi everyone online and hello. So today we have Dr. AJ Mellott with us with Ronawk. He will be telling us about this really actually cool way of doing cell culturing, rather a heck of a lot more efficient than the way that's been done in the past. So he's the expert. I'll let him talk about it, but welcome.
Yes, thank you so much. And good morning, everybody. I'm really glad that you're tuning in virtually. And for those of you that are here, thank you. Those at home, you're missing really good coffee and cookies this morning. So I am AJ Mellott, CEO, co-founder of Ronawk. And I am really thrilled to be here. I have to give a special thanks out to Professor Prasad for the meeting and helping with this. And of course, Dr. Porter. So today, what I'm going to get into is how we are going about orchestrating cell behaviors in our novel 3 modular system, which is our core technology here at Ronawk. So my talk is going to be split into actually 3 parts. So the first part is going to be an overview over orchestrating cell behaviors. The second part is going to be about our core technology, the Bioblocks. And then the third part is going to be about our company, Ronawk. So let's get into it. So my background is actually in tissue engineering. I've been at this for 14 years. And traditionally, when I think of tissue engineering, the 3 pillars I was kind of taught were we have our scaffold, or we have our growth factors and our cells, and we use these 3 levers to manipulate how cells behave. But over the years, we've been able to explore new technologies that allow us to do even more. So when it comes to engineering cells, thanks to CRISPR-Cas9 technology, there are so many genetic modifications we can do to either adult stem cells or iPS cells that give us a lot of things to try. Additionally, on the biomaterial side, the advancements over the last 15 years have been incredible. We now have novel chemistries that we can use to make a whole library of different materials. We can incorporate growth factors into our material, or we can functionalize surfaces to include different growth factors. And then furthermore, now we can actually tune the different mechanics of certain materials to actually apply signals to cells and change how they behave. Now, the part that I'm most excited about, that I'll go into a little bit more later today, has to do with the structural components when it comes to a scaffold or an extracellular matrix. So during my postdoctoral work, I spent a lot of time actually decellularizing different tissues and implanting cells. And what I got into shortly thereafter, when I was starting my faculty position at the University of Kansas Medical Center, was actually additive manufacturing, or commonly known 3D printing. And so that gave this wonderful spatial control over how we could assemble different materials together. And so where this is important is when we want to influence how a cell behaves, there are certain parameters we have to take into consideration. So let's talk about shape and topography. So if I want to manipulate a cell, one of the first things I have to think about is the actual structure of what the cell is going to rest on. So when I have a material, no matter what it is, I need to think about, is that material going to be a solid? Is it going to be something that has pores? What are going to be the shapes of those pores, from a circle to a triangle to a polygon? How big are the pores going to be? How close am I going to put them together? And how am I going to arrange that? Because all those factors come into manipulating how a cell is going to behave. Furthermore, it's not just the surface, but it's also the topography. So the panel on the right is one of my favorites because, you know, when I think of cells, they're not too different from us. We are obviously just an amalgamation of cells. And so panel A is really nice here. If you think about, you're walking down the street, it's flat, it's nice, you can be all relaxed, you can kind of spread out. But then in panels B, C, and D in that right figure, what happens if you're walking along and there's either cracks or grooves on the sidewalk or other obstacles? It's going to change how you actually walk down the sidewalk, as illustrated here by the cells, how they're traversing these different topographies. And then even in the more extreme with E, F, and G, we can direct where a cell goes and how it moves by either creating channels like an alleyway, or we can put all kinds of obstacles in the way that the cell has to navigate around, or in the worst case scenario, there are potholes everywhere and you are trying not to fall over. So once again, cells sense this and they have to navigate. Another component or another parameter that's really important has to do with the mechanical behavior of cells. In fact, cells are really sensitive to the mechanical cues. Think about, once again, using the sidewalk analogy, it's really stiff, it's easy to walk on, it's easy to balance. Try walking across a trampoline. That's why we like to jump. In this experiment, this was something that was really fun that I started my first year with my faculty position with medical students. And what we did is we wanted to understand cell behavior a little bit more. So in this experiment, we took parafilm, which hopefully most of you know is that nice stretchy lab version of basically saran wrap to seal containers. And we coated it with an ECM coating. We put mesenchymal stem cells on top. We let them form a monolayer, and then we took a 32 gauge needle and we punctured the parafilm. We let the cells grow for another 24 hours. We fixed them, and then we added a couple different amino-labels. Blue is for DAPI for the cell nuclei, red is mitotracker for the mitochondria, and then green is phalloidin for the actin inside the cells. And what we noticed is in 24 hours, the cells had gone from being a monolayer and formed this really tight ring around where our actual puncture mark was here in the middle. And it was fascinating to us that it happened that fast. We did a few other stains and we actually found that the cells were actually starting to secrete factors for contraction like smooth muscle actin, much like what occurs in wound healing, which is actually one of the focuses in my lab group. So then we took this a step further. When we got our first bioprinter, it was like Christmas morning for all of us in the lab. And so we decided to take this experiment and turn it into 3D. So what we did is we took a glass cover slip and we printed a donut on it that was made of gelatin methacrylate, and it was a 20 centimeter outside circle, a 10 millimeter on the inside, 20 millimeters on the outside. And what we did is we took our human wart and jelly cells, which these are mesenchymal stem cells you can get from the umbilical cord. We suspended them in matrigel, and then we actually put the matrigel inside of the donut hole on our glass cover slip. And then we exposed that donut to different formulations of media to affect the swelling of the donut on the actual matrigel. And so this created a concentric compression, and we were actually able to modulate a couple different secretions from the human wart and jelly cell. So all of that was just to illustrate the fact that mechanics are really important and a great tool for orchestrating cell behavior. So now we're going to get into the Bioblocks, and there's a little bit of a journey here with this actual technology. So nearly 10 years ago, I was sitting in the lab trying to finish up my dissertation, and I had this really awesome project where I was basically growing stem cells to genetically reprogram them to create sensory epithelium for restoring hearing. The downside of this project is that it required to grow a large number of cells, not just millions, but billions of cells. So for any of you out there that have ever done cell culture and know how painful subculture is, you're going to appreciate how difficult and challenging this part was. I spent hours in the lab, as have many technicians and grad students do every day all around the world. And yes, it's very annoying. There's a high chance for contamination. It's very monotonous. So in my frustration in doing this, I wanted to find a way to make this easier, make it better. And so, I started thinking, okay, well, why is it that we grow cells in 2D anyway? And so as I started digging into this, I realized, well, we grow cells in 2D first because it's easy. Plastic like this is pretty cheap. But here's the problem. When we grow cells in 2D, that's not how they grow in the body. In fact, there is nothing in your body that is naturally as hard as the plastic in this flask. Furthermore, there is nothing in your body that is this flat either. Everything is contoured. So that got me thinking a little bit more. If we're doing all this work in 2D just to build up to go into an animal model or some other preclinical model and testing and something fails, was it really worth it? What if we can do something that's more natural? Now, fortunately, I'm not the first one to have this idea. In fact, we have brilliant engineers that have been working on this for a very long time, and they have come up with wonderful systems for growing cells in 3D, from static suspension to hanging drop and forming spheroids, which are a wonderful tool today, spinner flasks for actually getting cells to perfuse, rotating culture vessels, magnetic levitation. Microfluidics has literally exploded in the last 8 years. Gel embedding and just making other gelatinous systems for spheroids. And so what's great about all these systems is that when we culture cells in these systems, we get functionally or physiologically closer in function to what we're studying in the human body. We get higher quality cells, and we can achieve more accurate results and reduce our reliance on actual animal studies initially. And so as I thought more through this problem, I wanted to build off of all the great research that has come before and try to make something that could at least incorporate these tenets and help researchers. And so here's what the initial idea was. So I'm sitting there and I'm thinking, all right, step one, I need to find a way to make a substrate that can be expanded. One of the problems when it comes to 2D culture is, once again, this is a discrete container. When we grow cells in this container, eventually they are going to reach the edges and then they're going to start growing on top of each other, and that's really bad. So we have to chemically dissociate them, take them out, split them into more flasks. Every time we do that, especially when working with primary cells, they behave a little less like the cells we need them to be. They lose some of their phenotype over time, and eventually they become senescent and they stop dividing. Not to mention, as you split flasks, you increase your risk for contamination because you go from 1 to 2 to 4 and so on and so forth. So if we could expand it, then we don't have to traumatize the cells by dissociating them. The second part of this idea was, all right, we've got to find a way to grow these cells in 3D. So the initial concept was basically inspired by Lego blocks, which who doesn't like to play with Legos? As I thought more and going back to what has been shown in the field, another thing I realized over the next couple years in validating the concept is there would need to be some sort of dynamic pore system or dynamic microchannel system incorporated into this scaffold. It's really important to consider how nutrients are going to diffuse and reach cells, how gas is going to be exchanged, and using vasculature as a model. In addition, as referenced earlier in the presentation, the shape of the actual microchannels is important for actually influencing how the cells would crawl through the structure and what that would do to flow and such. And then finally, the network arrangement within the structure. What kind of way did I want to arrange the channels? What would be most beneficial for the cells and transport, having them go all the way through, making some sort of grid, or some kind of sophisticated network. So all these ideas had to be incorporated into the design. And so now we get to, what is a bioblock? Well, as you see up here on a screen, this is a picture of our bioblocks. And for those of you watching virtually on the camera here, this is a hard plastic version of one of our bioblocks. It is a cubic centimeter. Yes, it does look like a jigsaw puzzle piece. What's really neat about this hydrogel is what we can do with it. So this is something we actually 3D print in our labs at Ronawk. And one of these little cubes here, one of these little bioblocks, can grow up to as many cells as you can fit into 120 of these T75 flasks. Just to put that in perspective, that is the max number of T75 flasks you can fit into a single CO2 incubator. So we've now compressed it all down to this one little bioblock. And here's what's really cool. When you need more room, you connect them together. And so it works really well. So I'm going to get more into this, and these are really fun to use. So from a business perspective, this product helps improve time to result. For those of you that know how tedious cell culture is, this makes it effortless. It saves you so much time. Imagine now only having to feed one well in a 6-well plate instead of dozens or 100 flasks in a city. And it increases opportunities or biological opportunities, and I'll get more into that here in a minute. So as we've said, one of these blocks can potentially replace all the cells in a CO2 incubator. When it comes to looking at the protein, the exosomes, and things that are secreted, we are getting comparable results to on a milligram per milliliter basis of what's in a stir tank bioreactor. And then finally, we can just further continue to expand your footprint of what you can do with cell culture without actually having to buy more lab equipment. And this already fits in all of the compatible cell culture vessels, CO2 incubators, and a lot of imaging systems that are already currently available today. So let's talk about the Bioblocks that we offer at Ronawk. We actually have 3 distinct Bioblocks that we offer. The first one is the E block, either for exosome or extra. This block is great if you want to culture a lot of cells, but you're more interested in the extracellular vesicles, the exosomes, the proteins, the other biologics that come out of the cells that you can collect in the media. You want the E block then. The T block is our original bioblock, and the T block stands for tissue. So the T block is actually the strongest of the 3. This one's great for if you want to grow spheroids, you want to make organoids, or you actually want to grow tissue in the hydrogel. And then what you'll do at the end is, this is the one block that can actually be fixed. It can be flash frozen. It can be either embedded in paraffin, embedded in OCT, and then it can be sectioned or cryosectioned for either histological staining or antibody labeling. So the T block is what you use for doing 3D physiological modeling before you go to an animal model. And then the final block, the X block, stands for extraction. And our X block is used for when you need to grow a large number of cells, and then at the end, you need to actually extract those cells for a terminal assay, such as RNA-seq, flow cytometry, or some other characterization assay where you need the cells in some sort of free format. And we provide an enzymatic reagent that degrades over 90% of our block. All you do then is strain the cells through a cell strainer, and you're good to go. You can pellet them, do whatever you need. And the best part is all 3 blocks fit together and are compatible with each other. So if you want to start with a T block and then you need to have some cells go to be harvested later, you connect an X block, you let them grow, and you can just keep propagating the actual culture. So how do you use the Bioblocks? Well, here's the thing. They're also super easy. So right now, there's only 3 to 5 steps, depending on what the goal of your experiment is. So step 1, which is optional unless you're using the T block, is you can coat the block. So you can add any type of ECM coating, such as fibronectin, collagen, laminin, or poly-D lysine to the block just fine. All those have worked. And then you can put your cells on. If you're using an E or an X block, there's no reason to coat. You can coat them if you want, but they're already pretty sticky. So then step 2 is actually seeding your cells on the block. And right now, the way this works is you're just going to seed your cells dropwise with a pipette, similar to how you would seed cells in a flask. It's super easy. You just work in a circular motion, and this afternoon I'll be providing a demo. Step 3, feed your cells. You change out the media on the same schedule you would as if they are growing in a flask. Right now, the blocks are agnostic to any media formulation. So once again, super easy. You just gently aspirate from the side of the well, not touching the bioblock, and then gently add the media to the side of the well. Step 4 is optional. If you want to expand your block, as we talked about before, all you do is connect the blocks together and you just keep going. And once again, ET, X blocks, all of them can be connected together. Then step 5 is harvest your block. If it's a T block, you can go fix it and then process it. If it is a X block, you'll add our reagent to gently degrade the block. And then the E block, you will just keep going, and what you harvest is the actual media to collect the proteins. So let's get into some of the data. One of our first experiments that we did was actually looking at whether we could grow cells without having to subculture and looking at their health. So in this experiment, what we did is we took mesenchymal stem cells, we seeded 100,000 on a glass cover slip, we seeded 100,000 in actually our T block, and what we did is we basically kept culturing the cells out to different time points, 15, 30, and 60, with the exception of the 2D culture. Since the goal was to not have to subculture, once the cover slip was 100% confluent, we went ahead and fixed and did our counts. But with the T blocks, we kept adding T blocks together and taking them out. And so at 15 days, we had somewhere in the ballpark of about 10 to 15 million cells. At 30 days, we had closer to 90 million cells across all our samples. And at 60 days, we averaged about 350 million cells. And so if you're looking on screen here, what you can see on the left side is actually the blocks at different time points. So in the upper left corner of the 6-well plate on the left, you'll see a block with cells growing for 15 days. As you go below that, that shows you a block that has been growing for 30 days. And then in the upper right corner of the plate is a block that has been going for 60 days. So you can actually see through the blocks. In another experiment, we wanted to make sure that the cells would actually migrate when we join substrates together. So in this experiment, we had our first T block. We coated it, same thing again. We seeded 100,000 cells in it. We cultured it for 5 days, and then we added a brand new T block, which is that one to the right that's kind of more of a white color. We cultured both blocks together for 2 more days, and then we fixed them, and we added immunolabels again. Same color scheme as before, which is something we like to do at Ronawk. Green for phyloidin to show the actin, red for the mitochondria, blue for the cell nucleus. And so what you see in panel B here is actually the different cells after 5 days that are just hanging out in our first block. But then in panel C, when you move over, all the black arrows are showing you cells that are right on the edge of the first block. And then the white arrow shows a cell that has actually traversed from the first block to the second. And for some of you in the audience that might be wondering about that gap might look big, but keep in mind, once these blocks were actually fixed, they shrunk a little bit in size. So the gap is actually much tighter and nowhere near as wide when you're actually culturing the cells. So let's take a few more looks at what happens with the Bioblocks. So here, the top panel is just to show how quickly liquid can actually go through the block. So this is not capillary action. The microchannels in the block are engineered such that the fluid freely flows through. The channels are quite large, so it's very easy for any type of media or fluid to go straight through the block. So over 2 seconds, we see with what was started here on top, starts to actually move all the way through the block in just 2 seconds. In panel B, underneath, this is giving a nice cross section of the block with confocal microscopy. So when we look in the interior of the block, what you'll see here is we have cells that have been seeded. They're nice and tight, forming what almost looks like a ball from this angle. As we move from the interior to the outside, going from left to right here, what you notice is the cells start to expand and almost look like a vortex. In fact, the ones on the right to me, I've always thought, if you like Star Wars, look like just before the Millennium Falcon goes into hyperdrive. And so What's really cool about this is these cells don't actually form a monolayer when they're in our block. They make these cool vortexes as they go into the pores, and they twist and they ball up, and they look beautiful when you stain them. That also creates some interesting points when it comes to looking at cells through phase contrast in our block, and we have guides to show on that, but it's different than what most scientists are used to. But it's also really fun too to see the cells behaving in this new environment that's more native. So then a few more pictures here. So in panel A, this is a 6-well plate, so we can fit up to 24 blocks into a 6-well plate. We seeded cells in the top left T block first. We let them go for 7 days, then we added 3 more blocks. And what's nice is in a 6-well plate, when you have 4 blocks, they actually hold each other together really well. And we cultured them for a few more days. And so then panel B is after everything's been fixed. We applied the same immunolabels as before. And now you can clearly see the top, or the first block that was seeded as the top portion. And then where the arrows are is showing the seam of the bottom of the second block, and cells have already migrated in. In panel C, we have a really nice picture of a close-up of one of the vortexes. And then in panel D was a mitochondrial stain, just looking at cells throughout the block. So one of the questions that's come up multiple times and that we were curious about in my own lab is, okay, that's great. You can seed these cells in these blocks. You can fit them together. You can eliminate subculture. But what does that actually do to the phenotype of the block? So what we did is we used CD markers that we immunolabeled. And once again, this is with the T block. So we were looking in situ. We couldn't actually extract these cells. And we applied the most common CD markers associated with the mesenchymal stem cell type. And what's shown here is actually the positive markers. We're not showing the negative here. But what I want to point out is we did this over time, and we had to come up with actually some new terminology. And so what you'll notice is we have P2 in 2D and 3D, and for 3D we call it the passage equivalent. And what we mean by that is that when we take the same cells and we grow half of them in 2D and half of them in 3D, when the cells growing in 2D have to be passage, that's when we call in our block the passage equivalent. So if it's 7 days for the first passage, then 7 days in the block is equal to the P1 equivalent. So we did that for P2, P6, P10. And what's great about the new X block now is, and what we're working on is now we can actually do side-by-side counts of cells because we can degrade the block. But with the T blocks here, the one that doesn't degrade, what we noticed over time is that as we analyzed the CD markers, we saw a higher expression of our stem cell markers here in the cells that were grown in 3D, and it actually maintained that number of cells longer over time in the Bioblock than it did in 2D. So fewer cells were expressing the positive CD markers over time as we went from P2 to P6 to P10. And that happens across all the markers here, and you can see pretty well here on the right where we start with CD73 and it starts to diminish over the passages. Same with CD105, CD90 and CD105. We then asked a really important another question, and this was something that one of our research assistants performed, and that was when working with primary cells and they senesce, what happens if we take the senescent cells and we try to put them in a bioblock? And we noticed something really interesting. We noticed the cells actually decreased the expression of senescent markers. So we decided to do this time-lapse study again, but this time using beta-galactosidase just as a basic senescence marker to see what happens. And so once again, at the P2 equivalent, we notice, and P2 in 2D and 3D, we notice senescence, overall senescence in both populations is about the same, right there around 5% or so. But as the 2D cultures get passage more and more, we start to see this dramatic change where the number of cells in 2D, the population that is expressing senescence is going up. And so by P10, nearly 25% of the cells that are grown in 2D are now senescent, whereas at the P10 equivalent, the cells in the bioblock have maintained the same percentage, right around 5%. So then this was something that we were excited about, and we had some questions. Okay, what's going on here, and can we leverage this for any type of wound healing? And so we work with keratinocytes in our lab, and keratinocytes, for those of you that don't work with them, they are the primary cells in your epidermis, your topmost layer of skin. It's what creates that watertight seal. And so we wanted to look at what the effect of ASCs grown in 3D could have on the behavior of keratinocytes. So in this experiment, we took adipose-derived stem cells from lipoaspirate from patients that donated essentially their fat after liposuction. And we basically we extracted and isolated the ASCs, and then we cultured them in 2D and 3D, and we started collecting the media from those cultures over time. And so we then cultured the keratinocytes with the conditioned media from the two groups on the keratinocytes, and we looked at a couple markers over time. So here we're showing metabolic activity via Presto Blue, and what we notice is the conditioned media from cells grown in 3D increased metabolic activity. When it comes to proliferation, same thing again. The conditioned media from cells grown in 3D increased proliferation rate. And then also of importance, the migration rate via scratch assay, migration of the keratinocytes was increased when they were cultured with the conditioned media from cells grown in 3D. So just to show you the morphology, I thought this was important to give a quick overview, but on the left here, we have keratinocytes grown in their normal keratinocyte media, and they're kind of round and bulbous looking. In the middle panel, we have keratinocytes that were cultured with the conditioned media from ASCs grown in 2D. And what's interesting about this is you notice there's fewer cells, and some of them have that bulbous rounded shape, but we start to see a lot of more spindle and fibroblastic shape. And then finally, the ASCs, or the keratinocytes that were cultured with conditioned media from ASCs grown in our T blocks are in the right panel. And while we see more cells, we start to see more of a heterogeneous population, which is really fascinating to me and something I want to probe a bit further, because that's not something I would think is normal. So then once again, we did our good old time experiment, and we looked at this over time from passage equivalents. And interestingly, we see, like we saw before, when it comes to metabolic activity with the keratinocytes, media taken from P2 or P2 equivalent, they have the same metabolic activity. But over time, as you take the media from cells grown in 2D and you advance the passage, the metabolic activity of the keratinocytes decreases, whereas the conditioned media from the ASCs grown in 3D remains relatively constant at the same level. And before I forget that, in case some of you are wondering, that dashed line you see in both graphs, that is the actual rate of metabolic activity and proliferation for cells, or for keratinocytes that were grown in their native keratinocyte media, so no conditioned media. A similar effect happens again on the proliferation side. Both conditioned media from 2D and 3D cultures on keratinocytes is about the same, but then over time, as we advance the passage in 2D, we see a decrease in the proliferation of keratinocytes, whereas our keratinocytes treated or cultured with the media from ASCs grown in 3D remains fairly constant. And just to show you again, here's what happened when we did our scratch assay. So keratinocytes that receive conditioned media are on the left from our 2D cultures. Keratinocytes that receive media from our ASCs grown in 3D are on the right. Once again, passage 2, we do a scratch in our well plates on the keratinocytes. The white outlines the scratch, and then this dashed black line in between shows space that hasn't fully filled in. And so there's not much of a difference between both groups when we're at passage 2 or passage 2 equivalent. We start to see a little bit of a difference at passage 6, but where it's really pronounced is passage 10, where The cells have migrated very little when treated with conditioned media from ASCs grown in 2D, and the wound has nearly closed with conditioned media from ASCs grown in 3D. In addition, we decided to start probing the content of exosomes. And so we did 3 different assays here. And what you'll notice in panel A here is across all 3 assays, Nanodrop, Bradford, and BCA, we see a substantial increase in the number of exosomes that were secreted and collected from our cells grown in 3D versus 2D. In panel B here, we're showing keratinocytes that were actually, the nuclei are stained in blue. The exosomes this time are stained in red, and then phalloidin again to show the actin. And the keratinocytes are actually taking up the exosomes. To zoom in a little bit more, what we have here is now you can see this interesting change where the keratinocytes aren't just taking up a few of the exosomes, they're really taking up a substantial amount. And here you can actually see with the actin, now it's actually kind of forming this oval shape around them, or crescent moon shape in some of these. So this is something we're getting deeper into characterizing right now, and we're hoping to present a paper for submission here by the end of the year. So then, besides the work that we've done in our lab, we've been really fortunate to have some spectacular collaborators around the world. And one of our collaborators over in Europe, Dr. Actis and her group, they actually grow liver carcinoma cells and make spheroids. And so they've been using our blocks to make spheroids. And so these are pictures that she kindly sent to us, and these were shown last year at the AASLD meeting in Boston. And so what I want to show here, she was using the T blocks. We actually gave her 3 different types of T blocks with 3 different stiffnesses, and moving from low to high magnification, going left to right. After 7 days, you can see in each type of T block we had beautiful spheroids forming with her cells. Furthermore, she even kept these going, and at 14 days, we saw, once again, still beautiful spheroids growing, and she's continued to move with these, and we're anxiously awaiting new data to see how long she's been able to grow these spheroids, because what normally happens is when you grow spheroids, they reach a point where you start to have necrosis form in the center of the spheroid, and everything starts to collapse in on itself. But these are going strong, very healthy. This is just for fun, a beautiful picture that she took in our block with green for the mitochondria, red for beta tubulin, and then blue for the nuclei. So then, as we've worked with other collaborators around the world and around the US, they have been fortunate enough to try all kinds of different cells in our blocks. And so here we have a list of a variety of different cells that have been cultured. And what we're really excited about and hoping with today's presentation is, as a company, we're small. We don't have the ability to actually test every single cell out there. But what we do want to do is we want to help accelerate your research. And so we're looking for collaborators that can try new cells we haven't put into the blocks and just give us feedback, share your data. It's something that helps us, and we want this to help your own research. Furthermore, you'll notice on here, and I'll get to this a little later, we've not only grown human and animal cells, but we've actually grown plants in these blocks. There's a lot they can do. So now I'm going to talk a little bit about Roanoke. What is Roanoke? So Roanoke, pronounced Ronawk, is a combination of the names robin and hawk. And when you take the mythology behind the robin and the hawk, you get message of healing. And so our whole goal at Roanoke is to accelerate research for scientists by simplifying biology. Our goal is to make the researchers the heroes in their labs and the heroes to their clients so that they can discover the next generation therapies. And that's where we're here to help, because when we work with our colleagues and we all work together, we get more done and we help more people. We were founded in 2019. I was still working at the University of Kansas for the Department of Plastic Surgery. And we started off with a really tiny office in Olathe, Kansas, which is a suburb of Kansas City. And that year was pretty special. We had our first trade show. We got a few people interested. And we got enough attention that we attracted some angel investors and family and friends. And by 2020, we had received enough investment that we were able to move from our tiny office into an actual manufacturing lab. And not only that, but I was actually able to leave my faculty position and go full-time into the company. I still maintain an adjunct role, but now I could dedicate all my efforts to Roanoke. And so Eagle Lab, which is shown here in the top row, is our wet lab for creating new materials. Falcon Lab is our production lab for making our bioblocks. And then the Owl Room is where we have all of our microscopy. And then, of course, we have our own decon and sterilization room and our own generator. And so 2020 was interesting because we moved in and then the pandemic hit and everything closed. However, we were very fortunate that we got moved in, and what we did for that next year, year and a half, is we were able to provide overflow services with the equipment we had in our lab to different universities in the area. What was great about 2020 and 2021 for us as a company is we were able to further refine the technology, focus on development, and at the end of 2021, we had 2 new Bioblocks to offer. Furthermore, we still got to interact with collaborators, and it was because of their feedback during the pandemic that we were able to do more research and refine the product to get it to where it is today. And then by 2021, that was actually a spectacular year for us. We got some more attention, more articles, but the coolest thing about 2021 is our staff grew. We went from just 2 individuals to actually 7. And once we got that team in place, this is where magic really started happening. We have barely gotten through Q1 of the new year, and already Roanoke has had its first webinar with the public. We had one of our state representatives come to visit us and take a tour, which was really exciting. We had a really nice article about us in Pharma's Almanac. But one of the coolest things that I hope gets everyone's attention is Biolabs at Pegasus Park opened, and we actually are a member of Biolabs here in the Dallas area, and we are working on setting up our office here. So we're pretty close, and more is to come on that in the future. So we're growing. And then, as I alluded to earlier, what's next? Well, I told you we can grow some plants in here. We're actually working on a new Bioblock that we hope to release early next year, but we've had some really exciting results with being able to grow some plants, and hopefully I'll get to work with more people, especially here in Texas, on the agricultural side. So as I come towards the end of my presentation, I want to thank our team at Roanoke, which is just an outstanding group of people. My co-founder, Heather Decker, is our Chief Technology Officer. I've been working with Heather for over 10 years. She is a wonderful individual. She's wicked smart. She's a microscopist, but it's thanks to her that we've gotten the attention, because she's the one that comes up with all the applications for the Bioblocks and finds ways to help scientists use them and make their life easier. Tom Jantz is our President and Chief Operating Officer. And the cool thing about Tom is he has spent a significant portion of his career in the startup world and has exited several startups and is just phenomenal at giving us that business side of experience that Heather and I didn't have. Scott Lee is one of our favorite people here because we've known him also for 10 years, and he has been in the life sciences industry for nearly 30 years. So when Scott came on, he really helped us on the sales side and the branding and just making us become a more mature company. And then with him, he brought Trice Alford, our Chief Marketing Officer, and Trice is just a superstar when it comes to marketing, online presence, and branding, and is teaching us new things every day. And then finally, my research staff from when I was at KU Med. So what you saw here, most of the data was actually performed by Jacob Hodge. So thanks to him and all of his hard work. The original hydrogels, the donut hydrogels that we tested with our printer, all that work was done by Brianna Hawthorne. And then Braden Stewart helped with some of the initial characterization. And so I've been lucky to be surrounded by wonderful colleagues in Kansas and Florida and also across the Pacific Ocean and the Atlantic Ocean. And I'm hoping to work with more bioengineering programs so that we can accelerate research together and do more good. So with that, I'd love to thank everybody for your time, and I would love to take any questions you have.
We have 2 questions online.
Sure.
This is from Shori. How did you ensure the shape fidelity of the Bioblocks during printing?
Ooh, that is a good question. So there was a lot of QA and QC that went into that. So we had to come up with our own designs. We had to work with software engineers. And then what we actually had to do in our own lab is use a microscope and different measurements to make sure every time that the block printed as it was supposed to be. And we still continue to incorporate other methodologies to test other quality control parameters. But great question.
Another question, Shori. What kind of 3D printers does the Falcon Lab have?
Great question. So we mostly use stereolithography printers. So those are printers where, for those that aren't aware, there's a couple different types of 3D printers. But with stereolithography, traditionally, there is a platform. The ones we use have a projector underneath. You will fill typically a Petri dish of some sort with your liquid polymer. A stage will come down, and what will happen is light will be projected up into the Petri dish where it will photo cross-link the gel so that it goes from a liquid to a solid. And then what happens is the stage raises with each defined section and resets. And basically you grow your object upside down. What's great about that is it's really fast. It's a really high resolution. The trade-off is you can typically only use one material. The other printers we have in our lab that we use for some type of prototyping are what are called fusion deposition printers. And those are probably what most people think of when they see 3D printers. Those have nozzles and there's an extrusion that comes out. And with the printers we have, we can combine multiple materials. And so the advantage with them is we can make objects that have composite materials, but the resolution is not quite the same, and they're also much slower. So those printers are great for prototyping. Scott.
This is Scott Lee. I work with Dr. Milan. We actually just got 2 questions through my email that get asked a lot, so I think they're relevant. One was, what is the physiological relevance of these cell culture clusters?
Oh, great question. So the question was, what is the physiological relevance of the cell clusters in the blocks, yes, or the blocks themselves?
The clusters in the block.
The clusters in the block. So when we have spheroids forming in the blocks, we can get a more native 3D arrangement of the cells. And so this is great for maintaining the behavior, the characterization of the cells in the block. And the reason it's important is because, so I'll give an example. With the wound healing studies we do some animal models. Typically, when we're looking at cells, we have to take biopsy from the tissue of the animal, and then we process it and we look at it on slides and stain it. With these blocks, we've created the opportunity, and this is only one of the early generations, but where we can grow spheroids. We can grow tissues that look like they do in the body for studies so that individuals don't have to use animals. They can get more information initially. And that's the biggest importance. As we move forward, we're hoping to be able to actually have bioblocks that are closer to bigger portions of tissue and not just spheroids and organoids, but more like a biopsy. And so it creates a starting point for studying morphology, physiology in 3D before going into an actual animal model.
Follow-up question to that is, what could be their applicability and how are these different from other cells grown?
Okay, the question was, what could be their applicability and how are the cells grown?
How are they different?
How are they different from other cells? So the biggest thing right now with these is when it comes to culturing most cells, that they have to be subcultured. We end up saving an enormous amount of time by having this mechanism where the blocks actually connect together. And what that effectively does is that creates an expandable substrate. And the cells can continue migrating. They actually don't know the difference on whether they were on a single substrate or a second because a second one's been added. And because they're growing in 3D, they can maintain a more native morphology. And so what does make this different from other types of cell culturing devices is, one, it's in 3D, so it's more natural versus 2D, like a tissue culture flask or a well plate. That's probably the most upfront. The next one is because we can grow so many cells because we're utilizing volume instead of surface area, we use less media, less reagents significantly. We've actually compacted 120 of these into a cubic centimeter, so that makes it more of a green technology. It also saves researchers an enormous amount of time. So instead of having to fill every 2 days, if I'm taking how most cells have to be cultured and fed, you're changing out media in a cell culture vessel like this every 2 days. And once you get up there, if you have a dozen, 20, that takes a lot of time. And then if you have to subculture, it's even longer because you have to dissociate the cells and go through that whole process. We wipe all of that out. And now all researchers have to do is change one culture. It takes 5 minutes and they're good to go. So they get all that time back. They can think more, they can do more analysis. When we look at other technologies, other 3D technologies, ours is the only one on the market that is expandable, an expandable substrate. Everything else is really good for modeling. This is actually meant to provide the end user with more options. So those are the big ones. And Scott or I would be happy to talk more for a more in-depth conversation on the differences.
We have another question. Do you think you can build these blocks using FDM or is STL the way to ensure accuracy?
I prefer to do STL on these for accuracy purposes. However, with FDM is great for giving us ideas of things that we might be able to try. FDM is challenging for production and mass producing, whereas with STL, that allows us to actually mass produce. It's a significant difference in the time scale.
Thank you very much. Okay.
Yeah.
It's always the lag, right?
Yes. So, and just before I forget too, I forgot to show this. If there is a question that comes up that I don't get to in the next few minutes, please feel free to reach out to Scott Lee. He'll either forward them to me or between him and I, we will answer those questions. But we love questions. We want to give you as much information as we can. As I said, ultimately with Roanoke and these Bioblocks, we want to help your research. We want to accelerate it. So that's what we're here to do. And I'm really hoping with this trip we can form some really cool collaborations. And if you're available, I hope you get to see one of our demos.
See any other questions right now? So thank you. I think we can wrap it up.
Thank you so much. Thanks, everybody. Have a wonderful day. Okay, great. Ready? All right. Good afternoon, everyone. I'm Dr. AJ Mellott with Ronawk, and I'm going to be showing you a quick demo on how to use our X-Block. For those that are joining us virtually and can see here, I'm holding one of our packages. This is the X-Block. it is a very fragile hydrogel, so hold the package from the edges. You'll notice that the side the label's on, there's a sticker here on the bottom to indicate that this side is the top of the block. The reason we notate that is when it comes to seating cells, you'll get the most efficient seating if the block is facing with the top up and the bottom actually down towards the well plate. Hence why we have that. So for the first step here, what I'm going to do is I'm going to show you how to take the X block out of its package and then marinate it with HBSS for 30 minutes. So in every one of our kits, you're going to get at least one of our X blocks or more, depending on what you order. Also, what will be included in the kit is one of our spatulas, which is sterile. The cool thing about our spatulas is this is to help you with handling the block. And also these spatulas can be autoclaved and reused. The X block cannot be reused, but the spatula can. So I've got everything prepared here. So first thing I'm going to do is I'm going to open up, take out my spatula so it is ready for me to use. What I'm going to do next is I'm going to take out, I'm going to empty some of the solution in my package. We have nice little tear tabs on top to make it easy. So we're just going to tear carefully from one side, and then I'm going to come over to the other side, tear back the other way. This is to help me with pouring off some of the liquid. And then I'm going to decant it into a waste conical tube here. And the reason I'm doing this is it just makes it a little bit easier to deal with the actual block and get it out of its packaging. It also helps so you don't get any kind of residual waste anywhere. There we go. What I'm doing is sometimes after you decant the liquid is it sticks a little bit to the packaging, so just trying to move it around. I'm going to grab my spatula here, and I'm just going to carefully lift the block out like so. And it's about the consistency of Jell-O, so you'll want to be very careful. And then I just slide it into the well. And for those of you that can see, now it's in the well here. If for some reason the block would flip on me and I can no longer tell what side is up or what side is down, what I recommend is take out a smartphone, turn on the flash, and take a picture of the block. If when you look at the picture and look up close, if you see microchannels and like a smooth white reflection, that's the top of the block. If, however, you see a rough side and all the microchannels look more dark or shadowy, that's the bottom side. So just in case you're having trouble if the block flips when you're taking it out, which sometimes does happen. So now what I'm going to do is I'm going to go ahead and I'm going to carefully add fresh HBSS and let the block sit for 30 minutes. And this HBSS is currently at room temperature. And this is just to help get the block activated and acclimated to room temp before we add our cells. So what I'm going to do is very carefully from the side, I'm just going to add my HBSS to the side of the well. I'm not going to touch the block. Sorry if this is not easy to see on here. I'll try to move my hands a little bit. And I'm going to need 8 mLs to fully submerge the block. So it's important that the block is fully submerged in HBSS so that everything can acquiesce to room temp and equilibrate. more here. And there we go. So what I'm going to do now is I'm going to go ahead and I am going to close this up. I'm going to set a timer for 30 minutes. And this is a perfect time while this is all marinating to go ahead and trypsinize cells for those to add. So I will go ahead and let this set to the side, and then I will be back in 30 minutes. Okay, welcome back, everybody. It's been just slightly over 30 minutes. So now that our block has been incubating with HBSS for 30 minutes, I'm going to go ahead now and I'm going to gently aspirate off the HBSS. And then today I'm going to be adding HEK 293 cells. So once again, I'll grab my pipetter. You can use a vacuum aspirator, but I recommend doing this manually just so that you don't accidentally touch the gel at all, the hydrogel of the Bioblock. And it's much, much easier. So I'm going to take the HBSS once again from the side of the well, being very gentle not to disturb the block. Then I'm going to tilt the block to get just the last little bit out. Or sorry, tilt the plate. And once I have everything out, I'm going to be all good. So here we go. Everything's been taken out. So now what I'm going to do next is I'm going to be adding HEK 293 cells. And so what we recommend is seeding cells at a density between 100,000 and 1 million cells. If you're working with a stem cell or a type of cell that is sensitive to contact inhibition because of confluence, you'll want to use lower to the 100,000 cells per block. If that's not an issue, you can aim higher for 1 million cells. Keep in mind that the cells will double faster. So with these cells today, I'm going to be adding 250 microliters for about 450,000 cells, just because we have a big concentration of HEK cells. Normally, when you add the cells to the block, you're going to want to shoot for adding either 250 microliters up to 500 microliters or half a mil. And what I will actually be doing here is I will be adding the cells dropwise to the top of the block, and then I'm going to let it sit for 2 minutes. Then I'm going to take up any of the excess liquid that went through the block, pipette it through again, another 2 minutes, do it a third time, let it sit, and then I will be adding the rest of my media to the well. So let me just adjust my pipette here. And actually, before I do that, I want to get everything mixed up, the cell suspension. So let me do that first. While I'm doing that, just to let you know, it's okay not to have any liquid on the block. You can go, depending on how humid it is, 5 to 10 minutes before anything will start to shrink. So it's okay right now. Let's see what we've got. We got a 200 here. Yeah, okay. We'll just use our 1000 pipette. And so what I'm going to do is just, like I said before, I'm going to add my cells dropwise to the top of the block. And I'm going to move in a circular fashion and just add it drop by drop. Okay. Hey Siri. Hey Siri. Hey Siri, set timer for 2 minutes. 2 minutes. So I'm going to let the cells sit in the block for 2 minutes. This is to help some of them distribute a little bit before I do my next pipetting of the excess liquid that went through and do it again. And what I'm trying to do is create some surface tension and get things spread out before I add the rest of my media, just so that the cells will be in the microchannels and I'll be able to stick and such. So in 2 minutes, I will pipette everything up again. All right, so 2 minutes is up. So now I'm just going to pipette up the excess liquid. And I'm going to repipette it through the block. So once again, this time I'm going to go the opposite direction and just add it dropwise. So the first time I went counterclockwise. Now I'm going clockwise. Okay. And so now we'll set this up again for another 2-minute incubation. Hey Siri, set timer for 2 minutes. 2 minutes, starting now. All right, we just finished another 2-minute incubation. So here we go. We're going to, once again, collect the excess liquid, pipette it up. And this time I am going to go counterclockwise, dropwise on top of the block. Being careful not to touch the surface and just add these drops very carefully. And now, depending on how much of a rush you're in, you can do that a couple more times if you want, or you can go ahead and let them sit. So I'm going to let them sit for a few minutes because our media is not quite warmed up yet. And then I'll come back in a couple of minutes here, no more than 10, and I will add the media to the side. So Okay, now that we've got our media warmed up and we've been letting this marinate for an extra 10 minutes, I'm going to go ahead and add the media gently to the side of the block. So I'm going to add a total of 7 3/4 mLs. So we'll start here. I'm going to pipette it out gently and just go to the side. And I don't want to touch the block. I don't want to pipette from the top of the block because my cells have been resting in there, and I don't want to accidentally flush them out. And so just gently add it to the side each time. The liquid will rise up through the bottom and the side of the blocks very slowly. And the goal with this is just to be as gentle as possible. Like I said, you don't want to touch the block. You don't want to disturb it. So don't hit the pipette plunger hard, just gently. Press slowly and add to the side of the well. And one thing that might happen as you get to the end and you're adding the last little bit of media, it's possible the block may float. That's okay. It's slightly buoyant. That's not a problem. So, and then after you've added your media, that's it. So you'll put it back in the incubator and you're all done. You'll feed the cells on your normal schedule. And then you can continue adding blocks to propagate the cells, or you can add our extract reagent if you want to degrade the block and harvest. And that's it. So thank you for watching. Have a great day.